Hi guys, this is Jonathan Feller and I'm with Rosalie Kiel, and in today's web lecture we are going to talk about comparative genome hybridization. This is short for CGH.
CGH is a technique that permits the detection of chromosomal copy number changes without the need for cell culturing. It provides a global overview of chromosomal gains and losses throughout the whole genome of a tumor, for example. There are two methods of performing a CGH, and both of them start with the test and reference DNAs, which are differentially labeled. The reference DNA is labeled green, and the tumor DNA is labeled red. The test DNA is partially digested to produce fragments. One of the methods is as followed. The fragments of both cell lines will be combined, denaturated, and pre-annealed. These fragments will hybridize efficiently to the denatured DNA to normal metaphased chromosomes. If you visualize the chromosomes by fluorescence microscopy, you can see differences in fluorescent signals of the test DNA relative to the reference DNA. A computer program will analyze the results and form a karyotyping.
The other method is array CGH. In this method, the labeled fragments are combined and added to a microarray and hybridized. This array consists of spots containing 100,000 base pairs of the human genome. The spots are analyzed by a computer and will light up yellow if there's an equal amount of green and red fluorescent signal. It will be red if there's an amplification of a locus, and it will be green if there's a deletion. Here you can see the results of a CGH. This diagram is a schematic figure that provides a color ratio profile of tumor DNA relative to the reference DNA. If there's an amplification, you can see the signal shifting to the right of the diagram, and the deletion will result in the shift to the left. To summarize this technique, the two different methods of CGH will detect changes in chromosome number and large structural changes. These structural changes include translocations, duplications, and deletions of specific regions. The diagram shows the specific regions in which you can see the signals representing genetic imbalances between the reference and tumor genome. The difference between traditional and array CGH is that array CGH is more specific. This was our web lecture from Jonathan and Rosalie. Thank you for listening and goodbye.